All right, so yeah, revealing the hidden diversity of Florida's mushrooms through DNA barcoding. Does anyone not know what DNA barcoding is? All right, one person, a couple people. All right, so DNA barcoding is basically just looking at a small snippet of the genome, a particular gene, at least with fungi, we use the ITS or the internal transcribed spacer. I'm not sure what you plant people use, but in fungi, it's the ITS for short. And the ITS does a really good job at delineating between species of fungi. So we focus on that for species level identification. Um, yeah, so I'm going to get into it. I also want to quickly plug the Florida Mycological Society. So this is a club we started last summer, last June in Central Florida. And every month, the first weekend of the month, we hold a mushroom foray. We go out, collect a bunch of mushrooms, bring them back, set them out on the table, and we've got field guides. And we go over identification and, just generally educate people on what it is they're seeing and if there's any potential uses or whether they're deadly or psychoactive, that kind of thing. So if you're ever in the Central Florida area or Orlando surrounding area specifically, first weekend of the month, feel free to join us. And yeah, that's us right there, and there, and some mushrooms. All right. So one thing I'm going to talk about a lot is a lab called Mycota Lab. They are in the Ann Arbor, Michigan area. So this is a lab run by my boss, Stephen Russell. And he has a project called the MycoMap Network. So what his goal is to outline the biodiversity of fungi in North America. And he's shooting for more or less complete, what's the word? I guess survey of species by the year 2035. And so what he does in order to do this is about 3 or 4 times a year, he holds these myco blitzes where anyone in North America can send in, usually it's like 10 or 20 specimens during a certain time period, usually like a week and a half to 2 weeks. And so he'll do like a spring, summer, fall, and winter one. And anyone in North America can send in their, 10 or 20 best specimens for free DNA barcoding. But on top of that, he has a select few regions slash states in the United States where he allows for unlimited specimens to be sent in. And Florida is one of those states, and it got recently added to this list at the beginning of 2025. So anyone here that lives in Florida and you find yourself out in the woods and you stumble across a cool mushroom, take a photo of it, put it on iNaturalist, take it home and dry it, and you can send that in for free DNA barcoding. So that's something I'm very enthusiastic about getting people to do. So hopefully one or two of you I can persuade. And yeah, if you want more information about this, like specific instructions, you can go to mycoda.com/the MycoMap Florida network. All right, so iNaturalist. Right now, so what you're seeing right here is a representation of all DNA barcoded fungi on the iNaturalist platform. So right now, as of yesterday morning, we're sitting at roughly 117K worldwide, which is a tremendous amount, especially considering only a year or 2 years ago, it was less than 50K. So, with the scalability, accessibility, and affordability of this DNA sequencing technology increasing, we have all this data coming in, and it's really cool. Right now in the state of Florida, as of yesterday morning, we have a little north of 2,300. And you can see all the observations that are DNA barcoded represented by these orange squares. So, in my opinion, forget about turning Florida red or blue. I want Florida to turn orange with DNA barcoded observations. Um, and you can see where we are here, Tallahassee, it's got a little bit, but the panhandle for the most part is pretty bare. So for you guys that live in this area, it's up to you to fill in the blanks here. Um, all right, so yeah, out of tw a little over 2,300 total sequence collections, Florida currently ranks as number 10 as of yesterday for the most sequenced observations in the US. And actually, back in September, I ran this list and we were number 9. We actually got demoted from number 9 to number 10. Missouri took our place, so we got to catch up to Missouri. Um, but out of those 2,300 sequenced observations, 1,293 of them have what we call temporary code names or provisional species names. So these are observations that we cannot associate with a previously described taxon or species. So these represent not 1,200 new species, but in total just 1,300 observations. And out of those 1,300 observations, we've created 323 new provisional species names or codes. So that represents potentially 323 new species that we've discovered. And this is all just in the last like 3 to 5 years since we've been doing this. So there's a lot of stuff to be discovered out there. And yeah, here's just a little bar graph I made of all the states, and you can see Florida right there, right behind Missouri. And then, of course, California, number 1. But all right, so next thing I'm going to get into is I'm going to go over the different ecological roles of these fungi and just share what I think are really cool examples of species that occupy these roles. So you've got sap. Does everyone know what a saprotroph is? No? Okay, so saprotrophs are organisms that basically eat dead organic matter. So when you see a mushroom in the woods on a rotten log, it's consuming that rotten log. That's a saprotroph. The next is symbionts or symbiotic relationships. So here I'm referring specifically to the mycorrhizal fungi. Does everyone know what mycorrhizal fungi are? No? Okay, so mycorrhizal fungi are fungi that have a mutualistic relationship with plants. And as far as the macro fungi go, typically it's host trees like pine, oak. So those are the symbionts. We've got parasites and pathogens. One a lot that's particularly what's the word, popular in like pop culture is these zombie ants. And they made a whole like show about it. I think The Last of Us, where you have these like cordyceps mushrooms infecting humans. But anyway, in reality, they infect insects and parasitize them and then create a fruiting body on top of them. So those are the parasites and pathogens. And then there's a whole host of species we don't know the exact ecology of. It remains unknown. So I'm going to start with the saprotrophs. So this mushroom right here is probably the most widely cultivated mushroom in the world. This is Psilocybe cubensis, also known as the magic mushroom. It's the easiest species of magic mushroom to cultivate. So most of the time when you come across psilocybin-containing mushrooms, you're going to come across this mushroom, but there are well over 160 different species of psilocybin-containing mushrooms. This one just happens to be very easy to cultivate, and it grows on the manure of ungulates like buffalo, cattle, horse, and maybe a couple other things. All right, so this mushroom here, Psilocybe niveo this one has a special place in my heart. This is the mushroom that got me into the DNA barcoding rabbit hole. So back in April of 2019, me and my buddy, at the time I was living in Tequesta, Florida, or right next to Jupiter, Florida, in Palm Beach County. And me and my buddy stumbled across these blue bruising mushrooms that were growing in wood chip mulch beds, like irrigated mulch beds, specifically in front of a security guard house to a high-income gated neighborhood. And we saw these mushrooms and we saw they were bruising blue. And one indicator or synapomorphy of psilocybin-containing mushrooms is that they bruise blue upon handling, and that's due to the polymerization of the psilocin molecule when it's oxidized. But anyway, so we stumbled across this mushroom, and me and my buddy, we at least thought we knew a lot about psilocybin-containing mushrooms, and we were familiar for what we thought was all the species that occurred in Florida that contained psilocybin. And this one didn't quite match any of them. And so, we kind of scratched our heads and we were like, what is this thing? And I eventually reached out to a buddy of mine, a fellow mycologist named Alan Rockefeller, and I sent him pictures of it. And I said, hey, Alan, have you ever seen anything like this before? And he said, no. I was like, well, what should I do? And he said, you should get it DNA sequenced. And at the time, I didn't know anything about that. So I was like, well, what is that? How do I do that? I had no experience with it whatsoever. And so he kind of, gave me guidelines, and I ended up sending it off to a lab in Spain and got a DNA sequence and got it back. And I don't know if any of you here have ever gotten something DNA sequenced, but when you get something DNA sequenced, you get back what's called a chromatogram, basically just this file with a bunch of different colored peaks that represent, A's, C's, T's, and G's. And again, I sat there scratching my head, like, what do I do with this now? And so I basically had to teach myself how to interpret and analyze this DNA sequence data. And so I finally put together a consensus DNA sequence, and I sent it back to Alan. I was like, all right, what do I do with it now? And I don't know if you guys are familiar with this sequence repository GenBank, but basically you can run any sequence through it and compare it against anything that has previously been submitted. And so we did that, and we found that it didn't match anything in GenBank. And Alan got back to me. He's like, yeah, this is a new species. And so I got really excited. I got like butterflies through my whole body. I was like, oh my God, new species of psilocybin-containing mushroom. These are mushrooms that people have been paying attention to for a long time. And, I'm sure a lot of you know there's a lot of hype and attention on these psilocybin-containing mushrooms as far as the therapeutic potential. And that really got me thinking, the ability to discover new psilocybin-containing species, this group of mushrooms a lot of people pay attention to. What else is out there? What other undiscovered fungal organisms are out there in the environment that we don't know about? And so at that point, I just started collecting every mushroom I could find and couldn't identify and bring them back. And at whatever rate I could, I would occasionally send them off for DNA sequencing. And yeah, I got addicted. I got hooked, and now I'm here talking to you guys about it. So yeah, this mushroom I can blame for sending me down the rabbit hole of DNA sequencing. Here's another picture of it. And so I mentioned that was in April of 2019. And then fast forward roughly 5 years later, in March of 2024, me and a couple other people officially published it as a new species, gave it the name Psilocybe niveo-tropicalis. You can see it's sort of a very light, like white kind of mushroom. Niveo in Latin means snowy or snow white, and then tropicalis, tropical snow. When you find it in these wood chip landscape beds, they tend to be, for whatever reason, in the dyed mulch beds. And so the white, in contrast, a lot of times with like black or brown dyed mulch, it almost looks like fallen snow at a distance. So I felt like that was the appropriate name. All right, Panaeolus oligotrophus. So this mushroom has an interesting story as well. So this one I started finding in my yard in September and October of 2023. I found it fruiting 3 times in my yard between September and October in 2023. And then, and I initially thought it was a different species that I was familiar with. But something you learn very quickly in mushroom identification is there's a lot of cryptic species, or meaning there's a lot of species that look alike macromorphologically, but are actually different. And so I had a hunch that maybe it was something different because the species I originally thought it was tends to grow in freshly laid sod with a lot of organic matter. And my yard is made up of basically just sand, and it's very weedy. It's not like a nicely manicured yard. Um, so I was like, okay, this is interesting. There's not a lot of organic matter here. It's not as nutrient rich as this species normally like. So what did I do? I sent it off for DNA barcoding, and lo and behold, it was not the species I originally thought it was. It was something entirely new. Now, I didn't find it in my yard again, but I eventually found it again in an abandoned citrus grove, which was basically just like all sand. And I found it again there. And then my buddy Aiden I think he's in the Naples area, he found it down there in like a pine scrubland. Again, just straight sand. So after those newer collections were made, I was like, okay, this thing definitely just like sand. Um, and it was, what's my next slide? Okay, here's some more pictures. Okay, so I eventually decided, because this is actually a genus I'm really interested in, the genus Peneolus. I decided I'd start describing it. And so in May 28th, I submitted it to be published. And then they accepted it. And then fast forward to October 7th, so just 2 months, just under 2 months ago, October 7th, I'll remind you, I haven't seen this species in my yard since September and October of 2023. October 7th rolls around, 8 a.m. I go out into my yard and I find this mushroom again for the first time in 2 years. Fast forward to 1 p.m. on October 7th, the publication went public and was published. So that was a little mushroom magic. It was, I see it as the mushroom's way of saying, today's my day. So I thought that was pretty cool. Um, this one's a species a lot of you are probably familiar with, Pleurotus jamor or dejamor. I'm not sure how to pronounce it, but this is the pink oyster mushroom. So oftentimes you'll go to the farmer's market and purchase oyster mushrooms in a variety of colors. One of them is the pink oyster. It's a more tropical species. A lot of people don't know, but we actually have this species here in Florida. Um, in this case, it's growing on a southern magnolia cone, which I thought was really cool because usually I see it on, the actual wood of the southern magnolia. So I decided to prop the little cone up on a branch and take some pretty pictures. Is there everyone here, has anyone not eaten oyster mushrooms or pink oyster mushrooms? Okay, a couple of you. Try them. They're really nice. Um, All right, here's another cool one. So here in Florida, we have all these palm trees, and there's actually a huge diversity of um, palm-specific fungi, and this is one of them. This is in the genus Clitosebula. So this is an undescribed species in the genus, and it grows out of. I actually learned this term recently, and I feel like I'm going to get checked now that I'm in front of the Plant Society. The old palm fronds, the old palms fall off and they leave behind those triangles. I recently heard they were called palm boots. Okay. So yeah, in these palm boots, you'll have all this organic matter like collecting in there. And so that's what this mushroom tends to grow out of. Like maybe not necessarily the debris in there, but it grows out from like the inside off the palm wood. That's like, it'll like grow out of the debris, out of the wood. And it always looks like they've got their heads kind of poking out of the boot. It's cool. Also, I don't have a picture of this in my presentation, but another really cool thing about fungi, and maybe for those of you who attend the walk tomorrow, will maybe get a chance to see it. But a lot of fungi fluoresce under UV light. So you can go out at night with a UV light and see a psychedelic forest come alive. And yeah, this mushroom will turn like bright blue under UV. And then while it's still attached, but once you pick it within like 10 or 15 seconds, it stops producing that reaction. So I don't know if it's dependent on being attached to like the substrate or if it's just got, a finite amount of fluorescent activity or reaction. But it's pretty cool to see. Oh, and yeah, that one, like I said, it's undescribed, but me and my buddy John Cook, uh, who helped start the Florida Mycological Society, we're currently describing it as Clitosobula persabili, so basically of sable palms. And this is just a really probably one of the most beautiful mushrooms I've come across. It was at Riverbend Park in Jupiter, Florida. If you guys have never been there, I highly suggest going if you're ever in South Florida. Just really beautiful place. But this is in the genus Pseudobaospora, and it's a genus that honestly, this is the only species I know of that occurs in Florida within this genus. Um, and I reached out to a guy in Italy, this Italian mycologist who studies specifically the tropical Pseudobaospora species, this guy Pietro Voto. And so I sent him some of the material of this species, and he's currently working on it. And he's going to be calling this one Pseudobaospora maculata, maculata meaning spotted. Apparently, when he looked at this mushroom under the microscope, there were blue and green spots in within the hymenium or like the fertile surface under the microscope. I'm not sure if it's strictly a chemical reaction to maybe the KOH he was using for the slide, or if it would be there in water or not. But he said it was really unique, and so he's calling it Pseudobaospora maculata. All right, this one. This is another sable palm associated species. This thing is like smaller than your pinky nail. It's like really, really tiny. The only reason I even saw this species was because I was looking at the white one, the Clitosobula, and I was taking photos, and I out of the corner of my eye saw these little cute little mushrooms, and so I was like, yeah, I'll take photos of these too. But this one might actually need a new genus erected for it, but the closest genetic match was the genus Gymnopolis, but it was still pretty far away. But for now, I'm just calling it Gymnopolis species Florida '04. Um, all right, so next we have the symbionts or the mycorrhizal fungi. And one of my favorite genera, the genus Cordonarius. Has anyone heard of the genus Cordonarius? Okay, so one of the more diverse mycorrhizal genera. So, I mean, worldwide, I don't even know, probably around 10,000 described species in this genus. Just super diverse and, a whole variety of host trees. Um, but I think they're really cute and cool looking. And Cordonarius, so um, little mushroom anatomy. So if you guys are familiar with the annulus or the ring you'll find around some mushrooms, after the mushroom matures and the cap opens, sometimes you'll see this like paper-like ring around it. So instead of a ring, they have this like cobwebby, spider-webby veil that attaches it, and we call that a cortina. And pretty much all the Cordonarius have this, and they also have rusty orange brown spores, and so usually this cortina will just be coated with the spores, and so usually it's this like orange brown color. But so here's a species I found in a hydric hammock by where I live, just south of De Leon Springs. And this one I'm going to be calling Cordonarius volusianus. Volusia County is the county I live in and that I found this in. So it's going to be named after it's called the type locality or the, original location from which it was found. Um, yeah, that one's really cool. And it grows under oaks. And for whatever reason, I always find I call them palm islands, in these like hydric hammock environments where it's flooded a lot of the time. You have these like raised palm trees where, the roots are kind of exposed, and then you have this like berm of moss around it. And yeah, for whatever reason, this thing I always find it fruiting from the moss on the palm island. All right, so here's another cool one. Does anyone recognize this mushroom? Yeah, the Mario mushroom, Alice in Wonder. Yeah, so a lot of you may be familiar with the taxon Amanita muscaria, which is the European sister species to what we have here in the southeast. Here in the southeast, we have Amanita persicina. You'll notice the cap color isn't as deeply pigmented red as the European variety. It's more like almost peach colored, or sometimes it can be almost pink. So we have this here in the southeast, and this one associates with pine trees. So this one is actually really interesting. The Amanita muscaria complex is both, or not both, but it's psychoactive, it's toxic, and it's edible all in one. So a lot of people will double boil or I've never consumed this mushroom, just so I can't speak to that. But a lot of people will boil this mushroom or double boil it or something and remove the toxins out of it, and they'll cook it up as an edible mushroom. And apparently, it's extremely delicious. People I know who have eaten it say it's one of their top favorite mushrooms. And then other people will, what's it called? Do what's called decarboxylate it. They'll like put it in an oven, bring it to a certain temperature, and I guess that converts some of the toxic compounds into psychoactive compounds. So the two compounds in it are ibotenic acid, which is the supposedly neurotoxic compound, and then muscimol, which is the psychoactive GABAergic compound. And I guess when you decarboxylate it, the ibotenic acid gets converted into muscimol, and it produces what's said to be an alcohol-like intoxication, at least in low to medium doses, and in high doses, supposedly, it can create a visionary experience. But this is different from your psilocybin-containing mushrooms. But yeah, I just think it's cool that it has like multiple functions and uses. You know, you can detoxify it, you can use it as a drug, or I guess the toxins in it produce like salivation and can also induce seizures. So I guess if you could probably kill someone with it as well. So it's sort of a three-in-one kind of deal. And then here's another species in the same section. So all the psychoactive, there's actually probably 100 different species of these Amanita that produce these compounds. Yeah, and they're all in Amanita section Amanita. Here's another one. This one is this. I wish I had a better photo of like just the top of the cap, but right now this is an undescribed one, and it has a beautiful yellow color. It's a lot smaller than the Amanita persicina or muscaria. It's like maybe that big tops. And it associates with the oak trees in central and northern Florida. I haven't found it in South Florida yet, but presumably it contains the same compounds that the Amanita muscaria complex does. All right, so here's another Cordonarius. This is going to be called Cordonarius foxii. So who here is familiar with Harry Potter? Oh, a couple of you. So you guys may recall there's a character in Harry Potter, a phoenix named Fox, and this is like Dumbledore's pet bird. Anyway, so this phoenix like turns to ash and then like and embers and crumbles to ashes and then is like rebirthed through the ashes. So this Cordonarius, and it's got warm colored plumage or feathers. So this Cordonarius species is actually associated with pine trees in post-burn habitats. So after a burn in like a pine forest, you'll see this mushroom come up in, like the charcoaly soil, and it's got the warm coloration. So I felt a cool name for it, an appropriate name for it would be Cordonarius foxii after this phoenix. I when you look at a lot of these species names, all these Latin binomials, sometimes they can be a mouthful and like unrelatable. And I felt like putting a little generational signature on one would be a cool idea. Uh, all right, so our next one, Inosperma ariaves, non-prov. So this one is in the family Anosabaceae, which is comprised of maybe 5 or 6 genera. And a lot of these species contain the compound muscarine, which can be deadly. So these are toxic mushrooms, although there are some species in specifically the genus Inocebi that contain psilocybin. But this one does not. This is another group of hyperdiverse mycorrhizal fungi. And so this one, I'm calling Ariaves, Ar Aria something, or Arius, I think means like golden, and then Aves, bird. So this is like golden bird. So part of the reason I named it that, it's got, this like kind of golden color. But this species is found throughout the East Coast, and actually during the summer months, you'll find it fruiting up north in like New York and Pennsylvania, but then it doesn't fruit down here in Florida until the winter. So it's kind of like a snowbird, these New Yorkers that come down to Florida during the winter. So I felt, golden bird was an appropriate species epithet. All right, here's another Amanita. This Amanita species FLO4, is in Amanita section phalloideae. Amanita section phalloideae is the section of Amanitas also known as the death caps or the destroying angels. So these are the deadly mushrooms, the ones you hear about, in news articles and old literature. But this is an undescribed one that I found growing right in front of an elementary school under pine trees in Jupiter, Florida. And it was funny. I was driving and I was gonna go visit my old job, and I saw these white mushrooms popping up on the side of the road. And so, me being mushroom fanatic, I make a U-turn, I park, and I go over there with my camera and I start photographing them. And there's this guy and his two daughters who are on tricycles, and they're on the sidewalk passing me, and he goes, "Oh, hey, what are you photographing there?" And I go, "Oh, these are some deadly amanita mushrooms growing under these pine trees." And I don't know if you guys have interacted much with people who are psilocybin evangelicals. I don't know if that's the right word, but people who are really passionate about doing psychedelics. This guy, as soon as I said mushrooms, he started telling me about how he cured his CTE with a super high dose of psilocybin mushrooms. And turns out this guy is one of these like famous athletes that has donated a bunch, like millions of dollars to MAPS, or psychedelic foundation. And anyway, we ended up talking for like an hour, and I was like, "Yeah, you've got these deadly mushrooms growing in front of your kid's school, so just be careful." Uh, he didn't seem to care. He just wanted to talk about magic mushrooms. But all right, so the parasites and pathogens. So this is an example of the zombie ant. And you can see here, so this ant is parasitized by this fungus, and then this thing coming out of its head is actually the fruiting body of the mushroom. So this is Ophiocordyceps campanoti floridani. And I forget the name of the ant species, but I know campanoti is in the species name for the ant. And I think there's a second species of ant here in Florida as well that this thing also parasitizes. Um, but yeah, so it's really cool to see. And these guys, you always find them either the Spanish moss on oak, and they'll just be like attached to the Spanish moss, or oftentimes I'll find them on air plants on oak. For whatever reason, it's always oak on either the air plants or the Spanish moss. Um, there's a guy in our club named Ben Tomerlin, and he is the ant cordyceps whisperer. And basically, every walk we go on, he finds them, and he even like carries around a little tube of them as like a party trick. He's just everywhere he goes, he has it on him, and he's like showing people this. Also, if anyone has questions at any point, feel free to shout them out, or yeah, if I need to clarify something, let me know. Um, the next one, the next parasite we've got is Talipocladium species New York 01. So this one was, I guess, originally found in the state of New York, where this temporary code name was applied. But since then, it's been found all throughout the East Coast. and it's down here in Florida. I haven't found, this is another one I haven't found in South Florida. I feel like the lower limit or the southern limit might be this Orlando area. But this one's interesting. So it might be difficult to see. So here's the Talipocladium fruiting bodies right here, these two guys. And then it's actually parasitizing a truffle in the genus Elaphomyces. So this is a mycoparasite, fungal parasite on another fungus. And yeah, it's pretty cool. I'm sure a lot of you are familiar with, these culinary truffles and, they're really hard to find because they're underground. But, these little Talipocladium species are little like little caution cones like, hey, right here. I actually don't know if this species here is one of these like delicious truffles, but it's cool. All right, so the unknowns. So like I mentioned earlier, there are a lot of fungi. We don't know the ecological roles that they occupy. Um, and yeah, here's a couple really cool ones. So this is in the genus Clavulanopsis. This one was just described in the last few months. My buddy Joshua Birkback, he's in Knoxville, Tennessee, and he specializes in the club and coral fungi. So this is one of the genera he focuses on. And historically, in the state of Florida, no species of coral and club fungi have been described. So for the most part, we can assume pretty much any species in the genus Clavulanopsis are undescribed. And so he went to town, and I was like his designated collector of the last 3 years, and I collected maybe 5 or 6 different species in the genus Clavulanopsis. And right now, he's currently putting together a monograph for this genus in North America. And he's describing, I think, 8 species in total, and 5 or 6 of them are from the state of Florida. So that's really cool. So we're about to, I think, within the next 2 weeks, it's going to be publicly published, and we're going to have 5 or 6 new species in this genus. But one of them he actually described earlier this year. Was Clavulanopsis manus is anyone not familiar with the Buddha hand fruit? It's like this citrus fruit that looks like fingers. Is that okay? Is anyone familiar with this one? A couple of you. So yeah, if you Google an image of this fruit and you were to turn it upside down and plant it slightly into soil, it would kind of look like that. So that's where this name comes from, Buddha's hands. It's really cool. All right, I think this is the first new Clavulanopsis I found. This one's called Clavulanopsis flammula, which means little flame. So within the subgenus that this one's in, this is on probably the smallest species in Clavulanopsis, subgenus Clavulanopsis. And, kind of looks like a flame. So little flame, flammula. But that's one that's going to be published in this monograph here soon. All right, the genus Hygrosabe. Has anyone seen these out on nature trails? They're usually just littered everywhere on trails, at least in my area. But the genus Hygrosabe, the common name for them is wax caps, and they have like a kind of waxy feel to them. But they're all these like brightly colored, typically warmer colored mushrooms, and oftentimes they'll stain black. And yeah, they're just really beautiful and cool and cute. And so this one is Hygrosabe conica Mississippi 1. I guess it was originally found in Mississippi, and it's in the Hygrosabe conica complex. Interestingly, there's a guy in Colorado, and it's not this species, but there's a Hygrosabe somewhere in Colorado that this guy, what's his name, Larry Evans collects. And I guess he experimented with eating them and found that they produced a psychoactive effect that was unlike psilocybin and unlike Amanita muscaria. So potentially there are novel psychoactive compounds within this genus that are unknown to science. And who knows, maybe they're psychopharmacologically interesting, and maybe when if some like analytical chemist, starts studying them and characterizes the compounds, maybe they'll discover some new receptor or something. But I do not advocate anyone go out and just willy-nilly experiment with eating random mushrooms. Um, all right, so another one, Clavulanopsis cancripes. So this one, canker, referring to cancer or crab, and then PDs refers to like legs or foot, I think. Anyway, I thought these looked like crab legs. So yeah, uh, and you can actually see, I don't know what species it is, but we got a little mycoparasitism going on here, a little mold on the crab legs. Probably don't want to eat those. Might make you sick. Um, oh, I guess that was it. But yeah, go above and beyond for science. Collect mushrooms, put them on iNaturalist, send them in for DNA barcoding. Um, this is how we move the field of mycology forward. And like I said earlier, like just in the last 3 to 4 years, over 300 putative new species just in the state of Florida. Currently, just in the United States, we've outlined a little over 11,000 temporary code names. So that's potentially 11,000 new species of macrofungi. Um, and yeah, there's just a lot to be discovered. And what else do I got here? That's me. Yeah. Any questions, concerns? Yes? So I live in DeLand, which is Volusia County. I'm like 40 minutes northeast of Orlando. Uh, but I have family in South Florida as well, and so sometimes I'll go down there and visit. Uh, I do collect stuff in Alachua near Gainesville, because I visit the mycology lab there with Matt Smith. But really just all over Florida. Yeah.
Like how, if we want to take a picture or make a collection, um, you know, what kind of unique identifier, like talk us through in more detail if we want to go out and, take a picture and send a sample in, what do we?
Okay, so first thing you do, find the mushroom. Second thing you do, if you have any ID app other than iNaturalist, delete it. Download iNaturalist. Um, yeah, this is something I iterate over and over, iNaturalist, iNaturalist, iNaturalist. Download the iNaturalist app. Anyway, you found the mushroom, you got your iNaturalist on your phone, you got your geolocation enabled, at least while you take your pictures so that we have that metadata, time, location. Take non-blurry, up-close photos of multiple parts of the mushroom. This is something you'll see in these Facebook ID groups. I'm sure a lot of you are familiar with this. You put up a picture of a mushroom and people start yelling at you and they're mean to you. And it's because you took a really bad, overexposed, blurry 20-foot away photo of a mushroom. So get a couple good photos of the mushrooms, top of the cap. The number one most important thing, if it hypothetically, if you had to take just one photo of a mushroom for identification, how the gills are attached, assuming it's a gilled mushroom, how the gills are attached to the stem. That is extremely important. That's more informative than usually the color or the top of the cap. That's the number one thing. So make sure you get a nice clear photo of how the gills are attaching to the stem. Ideally, especially with certain genera like Amanita. Amanita have a volval sac underneath the ground, almost this like egg-like thing that the fruit bodies emerge from. Ideally, you would kind of dig up the whole fruit body and get an image of the entire thing. Sorry, you got your photos, you make an observation on iNat, and iNaturalist has this, computer vision AI thing where it tries to identify your organism, and it's great for things like animals and, correct me if I'm wrong, plants. Uh, It's 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 probably better for plants than fungi, but And there are certain common mushrooms that are what does Josh call them? Overly charismatic macrofungi like Amanita muscaria. It'll probably ID that, but most of them, maybe if you're lucky, it'll get to genus. But usually just leave it at guild mushroom, the order Agaricales. It doesn't really matter as long as you put that it's in, the kingdom fungi. It's fine. So some mushroom nerd like me will come in. Hey, this is what you got. Um, so yeah, make the observation. Now, every time you make an iNaturalist observation, you get a unique iNaturalist number associated with it, and it can be found in the URL. Although on the apps, and I know it used to be the case with Apple phones. It didn't show you this, but I think with their newer iNat app, you can now see this. If you open the observation and you scroll down a little, it'll say iNat number or iNat URL, and it's a 9-digit number. And so what you do, you would take your photos, you would collect it, and make sure you don't mix your collections because it's a PETA, pain in the A. Yeah. So I like using a tackle box with the little cells, and I organize and separate my mushrooms. I'll take it home. If I'm like trail hopping, in between trails, I'll put my tackle box on the dashboard of my car. And here in the Florida heat, especially during the summer, it'll start partially dehydrating. But in any case, when I get home, I will put it in a food dehydrator. Usually between like 100 and 130 degrees is fine. Honestly, most dehydrators don't go high enough for you to like cook the DNA. Um, so yeah, just dehydrate it. Make sure it's like cracker dry because if there's moisture in it and then you put it in like a Ziploc bag, there's a chance it'll like get moldy or something. So dehydrate it, put it in a snacks. I use snack size Ziploc bags because most of the things I'm collecting are pretty small. Only occasionally do I collect something large enough that requires like, a gallon or, a sandwich bag. And then you would write the INAT number on it, and you can send either that entire collection off to the Mycota lab, or if you want to study the mushroom, you could just take a small split and put it in the bag and send that off. But if you're not trying to go down the mushroom rabbit hole and spend all your time in front of a microscope, then you can just send the whole thing. And yeah, I would encourage you to send as many as you can at a time. The less packages that lab receives, the quicker they can get through stuff and the quicker you can get your results. And I would say as far as, okay, so another thing, how do you know your mushroom has been DNA sequenced? So it's really important you legibly write or even type up a label. And actually, my buddy Alan has like a label generator for INAT. You just give it the INAT number and it prints out this like nice herbarium grade label. Anyway, um, so once it gets sequenced, it gets associated with the INAT number, and then someone like me, a sequence validator comes in, and I look at your DNA sequence and your observation. So I'm taking that macro morphology, the location, and then also the DNA sequence to make a determination about what it is you found. And I compare it with all the reference data in a bunch of different genetic databases. I take that into consideration. I take into consideration what your mushroom looks like, and I make a determination. Um, Nia found a new mushroom species here in Tallahassee. I'm going to call this Gymnopolis species FL05. Congrats. And I'll actually import that sequence data onto the INAT observation. So there's what's called observation fields, and you can create these like custom fields for like anything you want. And so we have some like DNA sequence specific ones. And so if you go on and this is also specifically if you go on the website, it won't show you all iNaturalist.org. Oh, yeah, my coda here, I'll just pull it up. But yeah, once that gets imported, you can go to your observation and you'll get a little notification about it as well. And you'll see the DNA sequence. You'll find a link to what's called the blast page, or basically this hyperlink comparing your sequence to all the other sequences in the database. And from there, you can teach yourself how to interpret that and try and get a better grasp on what's going on. Yes.
About a year ago. And the website generated on the homepage is as easy as 1, 2, 3. It's not. And so I worked for, I don't know how long, to figure it all out. Their website was in part taken down sometimes, and it didn't show the address to the other samples to. So I kept going back to the host to the webpage, and I finally, there was the address. So I captured that and I drafted the whole procedure and instructions, from downloading the voucher slips to mailing the package off. And I can make that available if anybody wants those instructions.
Okay. Also, another thing that I know has given people headaches, the voucher slips that he suggests printing out. So I don't know if he wants everyone to know this, but that's an optional step. Um, because that does take a while to, like, fill out a voucher slip for every single one. I usually, like I said, I put the INAT number on a snack bag.
Say a lot about
Right. Yeah. And yeah, honestly, for beginners, I highly encourage that because it gets you familiarized with, what to keep your eye out for. But someone like me who's sending in like hundreds at a time, I don't have time for the vouchers. Yeah. Um, any other questions?
It's just the printing part, some of they get enough specimens.
My understanding is no. So they did I forgot what country in Europe, but they did a study. I think it was over a decade with, chanterelle mushrooms. Yeah, so they had this plot of land, and every year for over a decade, they harvested every single fruit body of chanterelles in this space over, I think it was like 12 years or something. And every year, they no effect. Yeah. There, while that's true, so the chanterelles are mycorrhizal, so they have that relationship with the host tree. But what is damaging? Habitat destruction. I would say if you have a mushroom that, mm, let's say, and you guys know more about rare plants than I do, but let's say you have a rare tree or something and it's downed and you've got fungi growing on it. Maybe there's a unique fungus to that host and it's a rare host. Um, you would just, out of precaution, maybe not affect that and take as little as possible. Um, I don't know. Yeah, I would say case by case basis, use your best judgment based on the ecology and what you know about the environment. Hmm.
So one force there, so that they can not just like rise to the top of the tree, but to spread it that way as well.
Yeah, and yeah, there's an argument to be made that every time you collect a mushroom, you're helping it, distribute itself. Yeah, a little fairy dusting around the woods. Yeah. Yeah, no, that's a good point. And also, if you're inclined to get into the cultivation of fungi, something that's definitely sorely needed is taking some of these rare fungi and putting them into culture for long-term storage. Just to, because yeah, with all this habitat destruction, I know there's actually there's a new species in the genus Psilocybe found in New Zealand about a year ago, and someone found it, they collected it, and the next week that habitat turned into a Walmart parking lot. Hasn't been found since. But because it's a psilocybin containing mushroom, people cared a lot about it. So it entered the underground cultivation community, and now people all around the world are cultivating it. And it may never be found in nature again. Um, so, any other questions?
So if all these new species that are being discovered, I think they have not been noticed before because I mean, most of what you were showing looked awfully small. Or do they look very similar? And so it's really the DNA sequencing that shows that they're different or
All right, so this gets into a very nuanced conversation. So there actually have been a lot of mushrooms described in the United States over the last 100 years. And okay, so there's a couple things going on. Like on one hand, historical fungal taxonomists in North America have adopted a lot of names from Europe and applied them to species we have here in the United States. And so we're finding that things that, people from the early 1900s called the same thing as the European species, genetically and then microscopically turns out to be different and ecologically. But also, there are a lot of things, a lot of type specimens that were stored in Fungaria from the early 1900s. And basically, the rate that they get DNA sequenced, because a lot of the early descriptions of mushrooms here in North America, they're very skeletal descriptions, and you can't figure out what the heck they were talking about. And so the only way to reliably conceptualize the species they were describing is to do microscopy or DNA sequencing. And I was telling someone earlier, here in Florida, we had the only guy who, for the most part, who worked on species here in Florida, this guy named William Alfonso Merle. He described, I think, 1,453 species in the state of Florida. But the thing is, he did that from the 1930s to the 1950s. The fungarium did not have air conditioning. It was humid because of Florida. Um, and then it had insect damage. And then he was also drying them in like an oven at high temperatures. So his stuff is notoriously difficult to get DNA out of. Although there has been some developments recently to get like really small snippets at a time, like 50 to 100 base pairs at a time, and then basically stacking them together to, you know. So yeah, earlier when I said we have 323 putative, new species here in Florida, I would guess at least 50% of them are probably new species that have not been described. But my guess is a lot of them have been described, and we have just lost that knowledge of what they are because there was pretty large gap in there being just like no taxonomists in North America. There's like a lull in fungal taxonomy. And so all the experts sort of died and, the torch wasn't passed forward. But now we're just starting to, yeah.
um, regular mushrooms that mushroom. What about polypores? Also taking those?
Yeah, so I am what I would call an agaric elitist. So I like guild mushrooms. That's what interests me. There are some polypores I think are really cool, like the Ganoderma, the reishi species. They're beautiful. But yeah, yeah, you're more than welcome to send in any kind of fungi. We are looking more for macro fungi, so the mold in your shower we're not as interested in. Um, but if it produces a fruiting body, then you can send it in. And yeah, there are some polypore people here in Florida that are really into that and send stuff in. And we've definitely found new species. We found a new reishi species that grows on pine in South Florida. Um, Probably a couple others too.
But I pull up behind that next app, so it's still stuff on classic, I don't want to do that. But on the details tab at the bottom, there's the 9-digit punctuation.
Okay.
ID number. And so if you want to do this with the mushrooms, you have to go in iNatural and join the Michael Map project.
Yeah. So that's
Or operate to the Michael Map project. You should.
Yeah, you should. Honestly, I personally don't do that. I make it as easy for myself as possible, and you know, yeah, if you can follow the directions completely and like follow all the instructions, that's great. But I don't think there should be any barrier to entry, especially if you're not a computer savvy person and you don't want to try and figure out, you know, how to navigate iNaturalist and stuff. As long as you can get them dried and labeled with that 9-digit number, it's going to get sequenced no matter what. And then honestly, someone like me, I tend to do a lot of heavy lifting on iNaturalist, and when I see stuff I need to correct, like I can just, fix it for people. I do that all the time. Anything else? No? Yep.